45, 50.
You do?
Last time I had issues with the we're recording.
That's fair. Also,
Yeah, I think that's
Yep.
the
I had a girlfriend once who was like a real germaphobe, and I had a school, and she wouldn't let me near her for 2 months after that. Like she told me, go sit over there whenever I came over, and I would go sit over there. Like, it was a different time in my life.
just hear that like
It was a different time in my life.
yes.
It's close by.
It was a different time in my life. 6 years ago.
We're going to get started soon, so please take your seats. Can they see you?
Yes.
Oh, through there. Okay. So they can see me.
Yes, they can see you.
Okay. Good.
I think so.
Please take your seats. We're going to get started. Okay. We're going to get started. So it's my great pleasure to introduce today's seminar speaker, Dr. Monica Kersch Becker. Dr. Kersch Becker, she's a good friend and colleague. I have known her for many years, since she was a PhD student in Cornell. So she comes from Brazil, but did her PhD in Cornell, as I mentioned, in the Department of Ecology and Evolutionary Biology. After her PhD, she did a postdoc at the State University of Campinas in Brazil, and then took a position as an assistant professor in the university at the University of Alabama. And recently she moved to Penn State, where she has an assistant professor position of arthropod ecology. She started at Penn State in 2020, is that right?
No, January 2022.
Oh, there we go. So
6 months. Wow.
she just started there.
Yeah. 10 months.
And her research is on multitrophic interactions that are mediated through plants and the mechanisms underlying these interactions. And hold on to your seats because it's going to be a great talk. She gives awesome talks.
Oh
So
my God,
thank
so
you.
sorry.
The pressure is on. Thank you, Monica.
Okay, wait. Okay, thank you, Cesar, for the lovely introduction, and thank you for this great opportunity for me to share my work. So today I'm going to be talking about complex species interactions, moving from trophic cascades to ecological networks. So trophic cascades occur when predators limit the density of herbivores, kind of like what we see here, where they are limiting the density of herbivores. They are having this positive benefit to the lower trophic levels, to the plants. However, this simplistic view do not parse out direct and indirect interactions that are happening within this community. This means that trophic cascades, like what we see here highlighted, do not occur in isolation. So we need to embrace the complexity of food web dynamics occurring in real communities. And one way that we can do this is by looking at the multifaceted aspects of direct and indirect defenses of plants, or the functional traits of the organisms involved in the second and third and higher trophic levels. So this means that there is a great need for research to take this multi-species approach to better understand complex ecological systems in which these pairwise or tritophy interactions are occurring. So I'm going to talk about my research that I did during my PhD, where I revealed the importance of plant resistance to trophic cascades. And I'm going to walk you through this path analysis where I color-coded the negative associations in red and non-significant associations in gray, dashed gray. So the first thing we saw was that plant resistance did not have this direct effect on aphid population growth. So when we were using different varieties of tomato plants that vary in resistance to aphids. So we were using plants that aphids usually do great on them, and plants where the aphids don't do that well, so their populations will not grow too much. But when we have predators in the system, that interaction kind of changes, and that's what we were interested in. What happens with plant defenses, whether it actually interacts with the natural enemies to have this dual form of control, of biological control. So plants didn't have this direct effect on aphid population growth. We found, as we expected, that predator consumption reduces aphid population growth. So if you have the predators, they are eating the prey, they are doing what they are supposed to do. But the interesting part is this negative effect of plant resistance on predation. So as we increase plant resistant levels, and yes, the aphids were not doing that well, it's also hammering the predators. The predators start consuming a lot less aphids on those plants. And that's not driven by the density of the aphid. Sometimes we can say, it's like, oh, but what about the density? So it's not the density. It is the communication of plant and predators. So what we concluded is that plant resistance has an indirect effect on population growth by changing predator-prey interaction. And we found out that one of the potential mechanisms is that plant resistance, the ones that were enhancing the effect of the predator, those plants were emitting higher levels of methyl salicylate, which is a volatile attractant cue for these predators. But one problem that I have with this setup is that I lump all my natural enemies in one big chunk that I call predation. So I did not take into consideration potential changes in their interaction. So to improve our understanding of biological processes across scales and different levels of organization, we need to take a community-wide approach to studying these interactions. So we can actually take into consideration in the abundance, diversity, composition, and functional traits of the organism involved, so we can understand the outcome, not only the outcome, but also the strength of these interactions. And we can do this by taking advantage of all these new approaches in statistics, not being afraid of R, and chemical ecology, microbial ecology, and ecological network theory. And I'm going to talk a little bit about some of these approaches today. So I broke down my talk in 3 main topics, trying to answer how we can understand these multitrophic interactions a little bit more. So at first, we are going to take a network-based approach to explore the structural role of interspecific variation in plant defenses on biological control. Then we are going to look at evaluating the cascading effects of the functional traits of predators on tritrophic interactions. And then we are going to look at how shelter-building herbivores facilitate not only arthropod colonization, but microbial diversity on plants. So a central goal in the study of tritrophic interactions is adding realism onto the existing trophic cascade. So integrating food web ecology and plant defense interactions, or plant defense theory. So we have been asking the question how we can combine plant defenses and biological control agents to regulate herbivore populations for many different decades. Because what we are trying to do is manipulate crop traits to enhance the effectiveness of biological control. So we want our plants to be working in concert with the natural enemies so we can control our past populations better. And this kind of question has been in the literature for over 30 years, and I want to use a different approach to try to answer it. So let's take a network-based approach to explore how plant defenses can enhance biological control. So before I get into the results, I want to explain what is ecological networks. They explore the role of interacting species in structuring ecoevolutionary processes in ecological communities. So for example, we can see in this hypothetical network And the beauty of this type of analysis is that we can assign our nodes, which are the little circles here, as potentially species or individuals. So anything that you want to test. So for example, here we can have plants, so different species or plants with different defenses. And then we have potentially the herbivores. So those are the nodes of our ecological network. And then we have the links. So the links is the interaction that happens and how frequent that interaction happens. So the thicker the connection, it means that you have reported that interaction occurring multiple times. Every time you do a census, you are finding that herbivore feeding on your plant. So, and why is this really cool? Is because we can identify the important species at the community level or the node level. And we can use all these community approaches like modularity, connectance, nestness. But for now, we are actually going to focus on node level metrics, which are more important for identifying the species important in this network. So they are going to estimate whether some species are more important in maintaining the network stability, how close together everything is, because we believe that stability is going to let the system to be more resilient to disturbances. So we can have metrics at the node level, and we can assign each node to be either a species or a functional group or an individual. So the two metrics that we are interested in is degree and betweenness. Those are the names of the metrics. So degree is the number of interactions performed by each organism. So here, for example, the highest degree is this species here. You can see many different links coming out of that central species. So it plays a really important role in this network. And then we have betweenness, which is very important for network stability. It links different parts of the network that would be either isolated or not well connected. So the highest betweenness is these two species here. Without this interaction, you would have two completely separate networks. And the more intertwined your network is, the better it is for resilience and stability. So what we can use is that this network structure and dynamics have substantial impact on ecosystem performance, but a lot of the studies come from natural systems. There is a lot of, like a big chunk of the literature with plant pollinator networks. So evidence from agroecosystems tends to lag behind the network theory. And these network-based approaches can be used, and why I'm excited about them, because they can be used to design systems to maximize a specific ecosystem processes or services. For example, if you want to increase pollination or biological control, so we can understand how the network is organized, so we can potentially use this species for biological control augmentation. So we can identify the species that are contributing most to pest suppression, for example. And how we did this, so we have our interaction network. Here we reported how the interactions happening between the different species, and then we are interested in the ecosystem service. So we can have herbivore control or pest control here in the y-axis, and the number of interactions, which is the degree metric. I find that number of interactions easier to digest than the word degree, so number of interactions. And what we expect is that the more interactions the plants are having with natural enemies, the stronger the herbivore control will be. Then we look at the trophy cascade. Remember, the trophy cascade is when you are benefiting the plants or the lower trophic level. So we can have yield or plant biomass as our plant benefit. And then we correlate that with herbivore control. So what we expect is that if you are controlling the pests of your system, your yield or the plant biomass is going to be higher in the end. So this means that ecosystem services can be maximized through the abundance of these important species or functional groups. So I'm very excited to present our new data documenting the structural role of plant defenses in plant arthropod interactions. So I'm gonna walk you through this network where first I color-coded the two levels of plant resistance that I have. In red, we have the high resistance. Remember, those are the ones that aphids don't like that much. They don't do that well. Then we have low resistance in gray, which is where the aphids really enjoy feeding on those plants. Then we have the blue circles, which are the natural enemies. The green triangles, which are non-aphid herbivores. So the aphids in these systems are accounted for the size of the square. The bigger the square, more aphids that plant has. So you see this one doesn't have a lot of aphids, but this one has. So then we look at how aphid density and plant resistance are increasing the interactions with the natural enemies. So when we run the analysis, there are 4 species of natural enemies that are contributing most to aphid suppression. The variegated lady beetle, the pink-spotted ladybug, aphid midge larvae, and parasitoids. Unfortunately, we were not able to ID all the parasitoids, so they were lumped as a group here. But those 3 species and the group of parasitoids is what is contributing most to aphid suppression in the system. So potentially, these are the species that we need to either help conserve in the system or potentially use in biological control augmentation to increase predator biological control. The interesting part here is that flea beetles are also playing a role in this community. They are actually connecting all these plants here that are not being visited by natural enemies and connecting those plants to the rest of the network. Exactly the benefit of that, we are still trying to digest this result and think about it. Would love to hear some potential hypothesis for that, but that's one of the species that show up as being important in the system. So when we think about plant defenses, what kind of potential defenses these flea beetles might be inducing on the plants and how that might be affecting, potentially repelling even the natural enemies in the system. So extracting the information at the node level, remember that we had those 2 metrics, degree and betweenness. Then we plot the high resistant here in red, low resistant in gray, and we see that low resistant plants are interacting with a lot more natural enemies, as you can see here with the longer bell-shaped curve. And for betweenness, which is linked to network stability, we also found that low resistant plants are promoting network stability in this system. So when we look then at the benefit, our ecosystem service biological control here in the y-axis and degree or the number of interactions in your x-axis, we see that the plants that are able to have more interactions with the natural enemies are the ones showing the higher pest control. So more aphids are being controlled on the plants that are showing higher levels of interaction. And the low resistant plants are the ones that are having all of these interactions. So higher number of interactions promotes pest control. So what we can conclude in this system is that what we really want is predicting the strength of biological control, but that hinges on us understanding the higher levels, how plant defenses are affecting their interactions and how they mediate arthropod interactions or predator-prey interactions and the structure of these communities in agroecosystem. So winning the constant battle against pests is a key element to ensure sustainable food production. And today, a lot of pest management scientists are using different plant resistant levels and biological control in IPM. But sometimes we don't have enough knowledge to understand how the plant resistant is working in concert with those natural enemies. So what we found in this system is that high resistant plants have a strong negative effect on the third trophic level, on predators and parasitoids. This means that they exert a weak control, a weak pest control on plants, and that actually increases the density of pests on your plants, so the density of herbivores, because they are being released from top-down control. So the only control in this system is mediated by the plant resistance. In contrast, our low-resistant plants are actually favoring the natural enemies. They have these strong interactions because they are attracting more natural enemies to our fields, and they have a strong control on the aphid population, so a strong predator-prey interaction, and that reduces their density on the plants. So you have a plant resistant working in concert with the natural enemies. So we can conclude that plant defenses drive the arthropod food web structure, and a stronger biological control happens on highly interacting plants. The plants that are able to emit lots of volatiles to attract these natural enemies. And where we are taking this study is to really understand how these plant defenses are enhancing the natural community of predators and parasitoids. So one of the first things we are interested in looking at, and we did some of this work in the summer, but I still don't have the data, is that plant defenses will strengthen biological control by directly increasing the resistance and the attraction of natural enemies. So we want to use a type of plant resistance that the aphids are not going to do well, but that plant resistance is not, it's going to keep working in concert with the natural enemies. And then we are going to use that to create ecological networks to look at the role of these defenses in structuring the arthropod community, the plant herbivore natural enemy network. And we are collecting volatiles from all of those plants to see if specific volatiles in this system are interacting more with the natural enemies. For example, if we can find that methyl salicylate is this central volatile that is attracting a lot more diversity of predators than potentially other volatiles that these tomato plants are emitting. So we are able to use this metric to test some of these ideas. And the third part would be to extract the main natural enemies that the analysis highlights for us, that the analysis show us, and try to actually manipulate them in the system to see if we have potentially positive or negative interactions between the players in this higher trophic level. So because we are interested in these functional traits, that takes us to the second part of the talk, trying to understand how these natural enemies might be interacting. And for that, I'm actually going to take you to the Atlantic forest in Brazil, where I work with this super cool system, and I'm very excited that I can share that with you. So this is a glandular trichome bearing plant, Trichogoniopsis adenanta, from the family Asteraceae. And why I like this plant so much is because it has an arsenal of defenses. It has this super cool toxic pyrrolizidine alkaloids, and a lot of us, if you're not familiar with alkaloids, a lot of us enjoy alkaloids in the morning, caffeine. So these alkaloids are in higher content on flowers and young tissue, which is a clear example of optimal defense theory, that the plants are going to invest more defenses in the most valuable tissue. What are the most valuable tissues? The ones that are going to be related to plant fitness, production of seeds, flowers. These plants have glandular trichomes, as I said in the name, and simple trichomes. So glandular trichomes are, they contain several chemicals in this kind of droplet on top of the trichome. And if you guys touch a tomato plant and that gives that tomato plant smell, those are the glandular trichomes that you are rupturing when you are touching the leaves. And the one that I was really interested, biotic defenses. So this plant has a facultative interaction with spiders. And that's why the plant has all this arsenal of defenses, is to protect against predispersal seed predators. So it's a super cool system because you have these dipterans and microlepidopterans that are gonna ovipositing. Here is a flower head. They're gonna oviposit in the developing flower head. And here, where is it? Oh, I think this picture doesn't have the little guy. But you can have the ovipositing inside the flower head, and the larva is gonna eat the developing seeds or the ovaries of this plant. So it has a direct impact on seed set. And as I mentioned, it has this mutualistic interaction. And remember, mutualism is benefit for both players or partners. So the plant has glandular trichomes. In Brazil, we don't have a winter where the species go over winter, but we have a dry and a wet season. During the dry season, there is not a lot of arthropods around. So the spiders rely on the arthropods glued to these trichomes for food. In exchange for this food, they actually reduce herbivory. You can see one of these link spider eating a caterpillar. And here is one of the microlepidopterans that starts going inside the flower heads and eat the flower head. So you have these mutualistic interactions where both species are benefiting from the interaction. But we have different spiders in the system. So we have spiders with different functional traits. So what is a functional trait of a predator? A hunting mode. So we have this beautiful lynx spider, which is an active hunting predator, and the crab spider, which is a sit and wait predator. Both spiders love to hang out on the flowers. That's where they stay. They are not doing a lot of patrolling. So if you visit the plant, that's where you're gonna find them. And the lynx spider is the one that is going around the plant a lot more. And there is a lot of interest to understand how different modes can actually optimize predation in a system. And the way they work and the way they can impact plants is through different routes. So we can have the sit and wait that will produce strong behavior responses. So the sit and wait predators, they scare their prey. And their prey is going to respond to that scare. And they also change the abundance when they actually eat the prey. But because of these strong behavior responses, we say that sit and wait predators, they have a trait-mediated indirect effect on the plants because they are changing the behavior, not only the abundance of the prey. In contrast, the active hunting predators, they promote evasive behavior responses. Sometimes the prey can see the predator and will show some behavior responses, but that's not as strong as it is in the sit and wait predator. So a lot of the effect that these predators have on prey and then cascading down to the plant is through reducing the abundance or the density of the prey. So they have density mediated indirect effects on the plants. So because of that, there is a huge interest to understand how the diversity of species can actually promote this stronger control. So adding more species with different functional traits can actually be good for the plants. But we need to remember that sometimes it's not the diversity per se, it's not just adding a new species, but how they are partitioning the resources, how they are interacting, that is going to affect the strength of that diversity. So a key challenge for us is to identify what are the important functional traits and how they are interacting. And as I mentioned, hunting mode will promote these differences in the predators. So we are going to try to focus on that. So the first thing we did was to look at the community-wide approach to predation risk. We did this by using non-lethal spiders and we placed them on the plants. What are non-lethal spiders? Those are dead spiders. So we caught them, froze them, put them back on the plants. So you can see here, those are 2 dead spiders, and we pinned them to the flower head. And then we have plants that there were no spider, just the pin to control for potential, flower stabbing effect. But what we were interested to see if the different guilds in the system, pollinators, herbivores, parasitoids, if they can recognize that there is a spider on the plant. Remember, the spider is not trying to catch them, The spider is not moving, it's just there. And you can barely see it, right? Like you can look at this, you don't even see it that well. So we look at the number of insects visiting. So we placed a camera and we recorded how many insects were visiting, sometimes when they approach and leave. So we took that all in consideration. So pollinators in the system, they didn't really care if a plant had or not a spider on them. So here you can see white, no spiders, red, the sit and wait, and gray, the active hunting. When we look at the herbivores, what we see is that they visit a lot more plants that didn't have spiders. So the herbivores, remember the predispersal seed predators, they were definitely avoiding the plants with spiders. They did not discriminate the two different species though. And same thing for the parasitoids. So the parasitoids are parasitizing the herbivores that go inside the flower head. So they also avoid the plants that have the spiders. However, I need to highlight that this is not controlled by density. So if the herbivores are avoiding the parasitoids, I believe they are using the cues from the herbivores, and that's how they are finding and deciding where to oviposit. So I don't know how much impact the spiders might have on them if I definitely control for the density of the herbivores. So, but what we can conclude from this is that we really need to take a community-wide approach when we are trying to understand predation risk. A lot of the predation risk studies, they tend to focus on a specific plants or a specific organism, but we can see here that different guilds are responding differently. And we can think about how much species-specific responses are also happening there. So then we decided to take those two spiders and investigate the sources of contingency in the strength of spider-plant interaction by using spiders with different hunting modes. And I want to recognize that I did this work with 3 amazing undergrad students that were working with me when I was in Brazil, Guilherme Pinheiro, Thaís Silveira, and Bruno Grisolia. And they all helped me with this project where we use a substitutive experimental design. What is that? So that means that we kept the density of the predators the same, we just changed the identity of the predators. So either plants with no spiders, plants with 2 individuals of crab spiders, and that's kind of the regular density we find, so they're not highly abundant on the plants, but the plants are also not that big. 2 individuals of the link spider, and 1 individual of each one of those spiders. Then we collected the flower heads because we were interested in that main group of herbivores, the predispersal seed predators in the system. So the first thing we found almost 400 predispersal seed predators. You can see some of them here. And in this NMDS, where highly similar community will cluster together. So we can see here that all the communities that had a spider, so sit and wait in blue, the active hunting in red, and in purple we have the 2 individuals of each one of the spider. We can see that it's all a big cluster here. We don't see a lot of difference. So it means that the community of predispersal seed predators is very similar in all these different spider species. But interesting that without spiders, you have a completely different set of community. So you can see that no spiders, it's separate from everything else. So spiders are changing the arthropod community in this system. And as I said, they cause a lot of damage. So here is one example. You have the pupa of these insects, and you have them damaging the seeds of these plants. When you don't have any spiders, a plant loses up to 60% of its seeds to predisperse to these seed predators. So what happens when we have the spiders? It drops to about 15% with crab spiders and about 30% with lynx spiders. So both of them reduce the density or the consumption of these predisposed to seed predators. And when we have both of them combined, it's kind of a similar effect with the lynx spiders. So we are not finding that diversity effect, right? Because we want that to be stronger than the two species by themselves. Then we look at how much those predispersal seed predators are consuming. So when you don't have the spiders, they eat about 25 seeds in each flower head. But that is reduced to about 7 for crab spiders and about 15 for lynx spiders. And again, when you have the two spiders, you have something in between, something very similar to the lynx spider. So you don't have like an effect of the diversity. But the interesting thing here, this is control for density. Remember that we have less herbivores on those plants. So we only focus on the plants that already had those herbivores or predisposed to seed predators. And what we see here is that just the presence of the spider is enough for them to stop eating and not consume a lot of those seeds. So this means that we have anti-predator behavior responses here. One of the main things that how herbivores are detected by predators is when they are consuming. They are moving, they are chewing, they are releasing defenses. So potentially there is a cost when they are, when the moms oviposit inside those flower heads. If there is a spider, the larva is going to eat a lot less. So that might have consequences for the performance of the insect as well. So then we decided to run path analysis, and I'm going to walk you through this path analysis because we want to take into consideration all this effect of the spiders and how it is affecting the seed set. Remember, trophic cascade. That's like our metric of the trophic cascade. So we use this structure equation model to evaluate the direct and indirect effects. So here we have, when you have the two crab spiders or the two lynx spiders. And they actually affect seed set through two different routes. So the first thing we saw, herbivores affect over-fertilization. If you have herbivores in the system, that's going to affect how much the plant can reproduce. This is an obligate out crosser plant. If it doesn't get pollinated, it doesn't produce seeds. So the herbivores arrive a little early in the system. So it means that the flower head is still not ready to be pollinated. So the herbivores, I believe they are probably changing the chemistry of these flower heads that will affect how much the pollinators can pollinate or their attraction to the plant. Spiders have, both of them have a strong effect on herbivore abundance, and herbivore abundance has a negative effect on seed set. So if we think about the indirect interaction here, is that predators have a positive indirect effect on seed set. And that indirect effect is through reducing herbivory. Both of them enhance over-fertilization. So that actually is telling us that they are not eating the pollinators, or at least not the important pollinators in the system. And over-fertilization, as you increase that, that has a direct effect on seed set. So we didn't detect any fitness costs associated with having these two spiders, even though people argue, oh, they can eat the pollinators. Yes, they can, but that's not affecting the seed set because their benefit in reducing herbivory outweigh the cost of potential consumption of pollinators. So what happens when we increase the diversity? Again, herbivore has this strong effect on over-fertilization, and the spiders reduce herbivory. But now herbivory doesn't have anymore that direct effect on seed set. So we don't have the positive indirect effect of spider diversity on seed set. We also lost the effect of spider diversity on over-fertilization. So it means that the spider diversity doesn't have more that indirect strong effect. And this actually suggests that there is fitness costs when you have these two species on the plants. And we link that to some negative interspecific interaction. If you remember our GLMs, our bar graphs, you always, every time that you have the two of them, it's never like the sum of both of their effects. This means that we believe that the crab spider gets really bothered by the presence of the lynx spider. So this intra-guild interaction is actually important because it's reducing the effect of the predators in the system. So taking into consideration this, it's important in the system. So we can conclude that spiders enhance plant fitness through distinct routes, either by reducing herbivory or increasing pollination when they are with co-specifics. The magnitude of trophic cascade, remember trophic cascade is the benefit to the plant, is contingent on the functional traits of the predator. Crab spiders always had a stronger effect than the lynx spider, so the outcome of the interaction doesn't change. It's still a mutualism, but the mutualism is a lot stronger when the crab spiders are present than the lynx spiders. So the presence of multiple spider species may carry an indirect cost to this spider-plant association because of their negative interaction. So this means that the strength of trophic cascade is contingent on the functional traits and the interactions between these different species. So remember when I mentioned about our tritrophic interaction, when we expand to start taking and understanding the role of these other interactions in the field, is when we start seeing that sometimes some diversity might not always be beneficial to the plants. It might have other benefits, but maybe when we include other plants, maybe that's why they end up visiting different plants. So shifting gears a little bit and building on this idea that we need to understand these different interactions in the system, I want to start considering herbivores that not only are changing plant defenses or inducing defenses, but herbivores that can modify leaf traits. And we have these herbivores that create leaf shelters, and they change the structural complexity of plants. And what is their role in this plant arthropod community? We take us to the last part of my talk, where we are looking at shelter-building herbivores and how they facilitate arthropod colonization and microbial diversity. So shelter-building caterpillars are considered ecosystem engineers, just like beavers on plants. And they create new habitats that are used by a lot of different species. And it's very cool because there are at least 40 species of shelter-building Lepidoptera on oaks, just on oaks. And the leaf shelters increase the structural complexity of plants, influencing the diversity of insects on those plants and on those leaves. But then we actually ask the questions, can they actually also facilitate microbial diversity in the phylosphere? So microbiomes are important for host health. We have been starting to talk a lot about this. And we have the phylosphere microbiome, which is the above ground microbiome, the microbiome on the leaves. And we have the rhizosphere microbiome. And it has been argued that the rhizosphere microbiome has a lot more diversity, and also that promotes higher functional diversity, or the abundance of functional groups as well. In contrast, the phylosphere has lower diversity and lower functional diversity. And a lot of the studies have been focusing on the difference between host genotypes or abiotic variation affecting the microbiome diversity. But I am actually asking the question, how does non-microbial organisms like insects might be impacting phylosphere microbiome? We know that a lot of insects transmit a lot of diseases. They transmit different viruses. They might be benefiting bacteria on entering plant cell walls by chewing on plants. But what happens when their visitation? Can just visiting a plant affect the microbiome of these plants? But these arthropod-plant microbiome interactions are largely unexplored, especially when it's not vector-borne diseases. What about these commensal microbes, or just herbivores or predators facilitating microbial diversity at the plant level? So we decided to carry out this experiment, and I did this as a lab project with my graduate students, Jake Hirschberger, BJ Subati, Danilo Borges, and my postdoc, Tori Kocius. And what we did is that we selected 15 trees, 15 pairs of oak trees, a white oak. And these trees were 15 meters apart, so you have one that would be the control and one would be the treatment. And what we did is that in each plant, we randomly selected 5 leaves. So 5 leaves for control plants and 5 leaves that were shelters. So how we create this shelter, here you can see the shelter. We roll the leaf exactly how a caterpillar does, roll the leaf, and we secure it with hairpins. So our control plants just received the hairpins. Then we waited for the ecology to take its course, and we collected those leaf shelters and the control leaves 10 days after we created these habitats. Then we recorded the arthropods, and to microbiome profile our leaves, we actually swabbed them. And then we performed 16S Earth microbiome pipeline, which include Illumina sequencing and bioinformatics with bacterial taxonomy in CHIME 2. What we found in this was we recorded 108 arthropods, either on leaf shelters or control plants. And we found that leaf shelters promote a 300 increase in arthropod abundance and diversity in these leaf shelters compared to control plants. We also saw that the leaf shelters here, control in gray, again the NMDS, where the different communities are going to be clustered apart, or similar communities are going to cluster together. And leaf shelters in red, and you can see the centroid is very different. And yes, there is a lot of variation for especially control plants, but the shelters are more tightly related. So this tells us that leaf shelters are altering arthropod colonization. But can that be changing the microbiome on those leaves as well? So the leaf shelters have a 13% more richness, which we measure in OTUs, and we're also 15% more diverse, or harbor a more diverse microbial community on the leaf shelters compared to control leaves. We didn't see any difference for bacterial phylogenetic diversity. But let's look at their community. So you have different microbial community or a different microbiome, either if you are in a leaf shelter or in a control. So it was very cool to start learning a little bit about this, because we identify bacteria that are often present in the rhizosphere, so below ground, some that are already known players in the phyllosphere. But interesting, we also detected insect gut microbiome bacteria. So some of these bacteria are related to increasing plant growth, protection against pathogens. So Pseudomonas can be a pathogen in many systems. The species that we found, it's actually one that is a common player in the phyllosphere. Then others that are aiding in decomposition and nutrient cycling. Some of them are in the rhizosphere, but others are present helping decompose probably insects on the plants or even fungi. And then we have, for example, Wolbachia, that is very common in insect gut, and that's on the leaf surface. So I showed you how the leaf shelters are affecting the arthropods and how the leaf shelters are affecting the microbiome. But how those two things are related, because that was what I'm interested in. If you have a lot more arthropods, maybe that's why you have a lot more microbiome, because these arthropods might be transferring either on their body. And what we found here on our y-axis, we have the microbial diversity, and in our x-axis, the arthropod abundance. And we found that you have a positive correlation between arthropod abundance and microbial diversity. But that only happens in the shelters. So as you increase the abundance of arthropods in the shelters, you are increasing the microbial diversity. We found lots of spiders. And I actually decided to break that down, the arthropod, into the different guilds. And my first thought was, the spiders are going to be bringing like other organisms, they're going to be eating things, and that's going to be decomposing. I thought that would increase the microbial diversity. It turns out that it's herbivore abundance that increases the microbial diversity in the phylosphere. And that's not through consumption. That's just through visitation. They are not eating the plants. They are just inside the shelters. The shelters help them protect from the environmental stress. So that, I thought that was really cool. And then I started getting really excited about this idea and about this lower functional redundancy in the phylosphere, higher in the soil. And then I started thinking about this phylosphere and was like, okay, these herbivores are increasing the phylosphere microbiome, its diversity. So can we actually maybe think that these shelter building caterpillars may provide the toolkit needed for the plants to withstand potential climate change effects? We don't know anything about that yet, but that's what we start thinking a little bit, because these are ecosystem engineers. So what if they are giving benefits to the plants that are more than just increasing arthropod diversity? So what we can conclude is that leaf-rolling caterpillars can influence phylosphere microhabitat and affect multitrophic levels. The phylosphere microbial diversity definitely stems from arthropod colonization, especially the herbivores. And this might actually have potential benefits for plant health. When we think about our agricultural fields where we want to wipe out everything, all the herbivores, what if they are bringing important microbes that might actually be helping the plants? I don't think we know anything about that yet to say that that's a potential, but I just wanted to kind of put that out there for us to think. So multitrophic interactions can be crucial drivers of population and community dynamics in this plant-associated microbiome. So some future directions in this line of research, and some of them we have started exploring, is either if there is a correlation between microbes and herbivore visitation. We can think about that different players in the system where we have pollinators, herbivores, or predators, and whether they are bringing different microbes to this system. And potentially if the microbes that are on this phylosphere might also be playing a role in attracting them. So what comes first, is the microbe or the arthropod, right? And does this actually have any benefit to the plant? So we need to actually see if this increase in diversity of microbiome actually has a potential benefit, either for plant health, seed set, biomass, anything. So right now, all we have look at, there is no benefit. But we didn't test anything like inoculating with a specific group. Sometimes like that can be a potential effect. And we haven't looked at all the different players as well. So we definitely want to see if potentially inoculating plants with this commensal bacteria can actually affect potentially plant defenses and prime them to withstand herbivory from other organisms in the system. So to finalize, just the take-home message from my talk, and I believe that is also the consumption rationale that motivates my research, is that these complex organism interactions are critical for insect population processes and community structure and ecosystem services. And what I really want my lab to contribute is to understand, to contribute to sustainable agriculture by shedding light into the role of these complex interactions, because we expect that these highly interactive and functionally diverse systems are also going to be more resilient when climate change starts really hitting hard on some of these plants. So with that, I would like to thank my students, collaborators, my small lab. So we just moved to Penn State, so I have only 2 students, but this is us collecting tomatoes in the summer. And with that, I'm taking any questions. Thank you.
Before we take from the floor, we've got one question from the chat, so I'm just going to read it off.
Okay.
So Cheng Lu asks, in your ecological network diagram, you said some species are more important because they connect between networks, i.e. increases the betweenness. In practice, how do you predict or calculate the importance of a species in that network?
So it is when you run the model with the bipartite packet in R, and it highlights kind of almost like a discriminant analysis, that it gives you some of the species that are contributing both, because your response variable is biological control or aphid suppression, right? And then it highlights which ones are the ones that are having a higher degree in that network. I think there is a question there.
Hi, okay, that was such a great talk.
Thank you.
In the first part of your talk, you were talking, like an interesting result you found was that the more defended tomato plants negatively influence your biocontrol, and I'm curious if that was consistent between predators and parasitoids, or was maybe like more of the case with predators, I guess?
So it was stronger with predators than it was with parasitoids. The parasitoids, I think they were not as abundant as the predators, but I didn't know how to kind of try to replicate the experiment, because I didn't want to release the parasitoids, but they were arriving later in the season. So it's almost that I started having the predators, and then later in the season it shifted to having more parasitoids. And the data that I had from the parasitoids is that their biological control was not as affected. It kind of like the curve, let's say the bar graphs, the mean was also similar, but it was not significant. So I cannot say that it would have the same effect. Right now, with the data that I have, is that they were not as affected as the predators were.
Okay, thanks.
Yeah. Cesar.
Very cool talk. So as you know, I'm very interested in some of the things that you are looking at, and especially like with the resistance and the biocontrol aspect. One of the things that we are interested is how the landscape affects these interactions. Mm-hmm. What's your take on it? Like, I feel like, would you see the same effect if you have different landscapes?
I think, like, usually landscapes, depending what is your surrounding, right? So if you are surrounded by areas that might be a refugee for a lot of these natural enemies, because that's one thing that people try to do, is try to have flower beds in the system so the ladybugs can actually eat something else when the aphid population goes down, because you always have that cycle, right? Like, it goes up, all the natural enemies arrive, they eat it all, goes down, then natural enemies leave, and then the prey comes back again. And I think when you have more complex landscape where you have these natural refugees or refugia around your crop system, maybe that going up, like, they will be able to arrive faster to the plants. I think that, like, and that would increase, like, if you think more monocultural landscapes compared to more complex landscapes. But that might also affect, like, how much you're using as your radius in your, because you use a buffer, right, like of 1 kilometer or 5 kilometers. That also might affect, I don't know how much it is the local effect with something that might be further away. Don't know if they would be able, because a lot of these chemicals is something that we talked about it too, like how far they will move to attract. Like it's something that you need to be really surrounding your field, or can it be 10 kilometers, miles away?
With your spider study, do you know why you didn't see any effect on pollinators? Like, that's kind of surprising. Do you think like the pollinators are foraging at different times?
So, it's interesting because the main pollinator is not a bee.
Oh,
So bees, they see very well for insects, and there are studies that show that bees discriminate the crab spider, like the way they sit on a plant, they can recognize that and avoid that plant. In our system, the main pollinator is a butterfly. Butterflies, they have those long legs. And the butterfly interacts with this plant because it sequesters the pyrrolizidine alkaloids, and the males present as a nuptial gift to the females the alkaloids that will be invested in the eggs. So it's a super cool, like tight interaction, like co-evolution part. And I think it is because the spiders cannot really hold. I think bees, they come like jump like this, right? Like belly first on the flowers. And then I think it's easier for the spider just to grab them. The butterflies, they come with their long legs and then they land in there. And I think the long legs is enough for the spiders not to be able to grab them that fast. So I think that's what I think, looking at the natural history and observing the system. I'm not sure if it is exactly that, but that's one of the things we discuss in the paper, like why we find this. Like but it was because bees were not important in our system. And a lot of the literature showing that predators, the spiders affect pollinators is usually bees. Yeah.
So we've got 2 more questions in the chat.
Okay.
oh, Kasia has.
Yeah,
So,
maybe.
we've got a question. Any reason that fungi were not included in the microbiome assessment?
No, there is no reason.
Okay.
Just cost.
Okay, that makes sense. Okay, all right.
But it is something that I was even talking with Cesar about it, that I think we will start including. I think we just decided to do bacteria. It was kind of like we wanted to see how this works, and something interesting came out, and that's how we are going to start building on that idea, I think.
Okay. The other one is asking about, could you explain a little bit more about the experimental setup? For example, how you monitored plant visitors. I know you did at one point say you used cameras, you know.
Yeah, so it varies. Let's say for my the tomato experiment, it was the going to the plant, squatting, looking, counting, moving to the next one. So it's just a census. I did multiple ones to increase my sample size. And the second one, in Brazil, we use a recording camera, and then we look at the videos, the recordings later, and we were able to, some things we're not, that's why we did by guild, because we were not able to ID species. Like, imagine those tiny little dipterans. We could see it because we identified the way they fly. It's different than the way the parasitoid fly, but they were just like dots.
It's tough.
Yeah, and the butterflies were easy. Those were easy, but butterfly, bees. So that's why we kind of broke it down into pollinators, herbivores, or parasitoids, because we couldn't get to species level.
Yeah, so my question was about the spider study that you did, and particularly about how when you increase the diversity of the predators, it didn't have an effect on seed consumption. And you made a comment about that potentially the lynx spider doesn't like the presence of
The crab spider doesn't
crab
like
spider
the
doesn't like the lynx spider. So did you see a decrease in the population? And then second part to the question, what do they do to the herbivore itself? Do they reduce its feeding while they're inside the plant itself, and that's what you're seeing?
So for the first part, when we did this, first, my postdoc advisor has been working in that system for like 15 years. And so he knew we had data from our natural observations. So the co-occurrence between the two spiders, it's a lot lower than it is when they are with co-specifics. So when we think about the natural colonization of, because these were spiders that we put it on the plants, right? And if you look at the natural distribution, I think it was like 20 or 30% you would find both species on the same plant. They were always spatially separate, and in our study too, they were spatial, like if a plant is kind of like this big, they would be in opposite branches. We were not putting them together. And I think it is just the presence because they eat each other too. So we don't know if they potentially might be eating their co-specifics less than they eat the heterospecifics. So maybe that's it. And what happens with the herbivore, a lot of them will just avoid ovipositing in a plant with spiders. That's the first thing. Once some of them do, the herbivore inside the flower head eats a lot less than the ones without spiders around. So I think how they are recognizing, how a larvae recognize a spider outside.
What would be the benefit of eating less as well? Because then you're kind of slowing your.
Oh, the spider can get inside and grab them from inside.
Oh my god.
Oh, so that, okay.
So if they are eating kind of on the top, they can like get in there. But I don't know. Like for me, I don't think that is something easy and that common. So I also don't know the potential mechanism of that interaction. But that was something we observed, and like I come up with the idea that okay, it must be like the predation risk, the risk of, because I don't see another alternative hypothesis for that. But maybe there is. But
Okay, great, thanks.
Please join me to thank Monica
Okay.
once again.
thank you guys. I can stop the
Yeah, stop.
share.
Stop recording.
Stop recording.